Thank you for joining us for this ECIS webinar dealing with cell invasion or extravasation assays. My name is Charlie Keese. I'll be your host for this about 25-minute long presentation. Let me begin by just briefly explaining ECIS, electric cell substrate impedance sensing. The basic principle of monitoring cells electrically was first reported when Ivor Gaver and I were at the General Electric Research and Development Center. There we realized that if you grew cells on electrodes and applied a weak AC signal, one could actually monitor some of their morphological changes. The heart of the system, of course, are the special arrays where there are electrodes. In this slide, we're looking at an 8-well array, which you can see in the upper left-hand panel. At the base of each well, in the center figure, you can see there's a small 250 micrometer diameter electrode. There's also a large counter electrode to complete the circuit that's labeled CE. But because the impedance of this is so much less than the small dot electrode, we essentially only see cell activity on that small circle. We can see a single cell on there or up to a confluent layer where maybe 50 or 100 cells would be involved in that small spot. Of course, that's a small sample, although there are reasons for looking at small numbers of cells. For most of the work we talk about today, it's better to have larger electrodes to get better statistical averaging. And so we sell a variety of different electrodes, both in 8-well and 96-well formats that have increased area to monitor the cells. But regardless of the electrode type, the measurement is much the same. Tissue culture medium is used as the electrolyte. The current will be carried by ions. The electronics provides a constant AC current source. It can be anywhere from 100 to 100,000 hertz in terms of its frequency, but it's always kept low enough such that the measurement is non-invasive. We, in effect, electrically eavesdrop on cell activities. We know the current that's being applied. We monitor the voltage across the electrodes, and by taking the ratio of the voltage and the current, we come up with the impedance, which is represented by the letter Z and is measured in ohms. But being an electrical AC measurement, it can be quite a bit richer. And let me explain that with this small panel. We're applying a known current, which is represented by the dotted red line. It's oscillating back and forth at whatever frequency we choose. The voltage, represented by the black solid trace, will have the same frequency. But what's important to notice is that the two curves don't peak at the same time. One would say that the voltage lags the current. The current peaks before the voltage does by a certain amount, which is usually represented by an angle phi in this case, thinking of the entire one cycle of the sine wave being 360 degrees. Well, one doesn't have to think much about that because the electronics take care of all of this, but by monitoring the amplitude and that phase angle, instead of just reporting the impedance, the ECIS instrument can report the pure resistance that's measured as well as the capacitance, as there is capacitance in the system from the gold plate electrodes, and of course the cell membranes also have capacitance. So that's what we measure at a variety of frequencies. Here's some typical data. This is on one of these small 250 micrometer diameter electrodes. We have two cell types that are attaching and spreading. As they do that, the resistive portion of the impedance, in this case measured at 4,000 hertz, goes through a striking change, going up maybe 7-fold in the case of the BSC cells in the first 2 hours. The NRK cells also attach and spread, but do so more slowly. But notice without cells, the lowest trace, it's absolutely a flat line. And so the fluctuations that you see in the curve that are shown here, which we term micromotion, actually come about because we're looking at such a small cell population, and those cells are moving randomly, even in the confluent layer, causing that signal. And other webinars talk about that signal and its importance, but it's not something we'll deal with today. Now, the use of larger numbers of electrodes, in this case we're comparing a single electrode and 40 small electrodes, results in getting rid of the micromotion, which sometimes is an issue and gets in the way of seeing the overall trends in the impedance change. Here we're looking at time zero at confluent layers of cells on either the 1E or this 40 electrode array. We've divided everything by its starting value. And so the resistance here all comes out at the ratio of 1, but that is a confluent layer. Shortly into the run, fresh medium is added, and that causes a change in the resistance as the curves both go up about 30%. Notice, though, that in the case of the one electrode, although the same trend is there, it's pretty much obscured, or can be at least, by the micromotion. And so for many measurements, having more cells and better statistical sampling is very important to see the smoother trends in the curve. The reason we see these changes is on the next slide. Again, we have a gold film electrode that's delineated by an insulating film. Current is carried by ions. It's a constant current measurement. So even when the cells go down and attach and spread, and some of them end up on the electrode, the current still is remaining constant. And now it has a difficult path, which causes the increase in the impedance. If we look at the solid lines, which come out from the electrode beneath the cell, between the electrode and the basal membrane, the current has to flow in that narrow cleft under the cell, and then ultimately flows between the adjacent cells in what is called the pericellular space, or the true barrier function of the cell layer. That's how the ions flow. But remember, it is an AC measurement, and so we can charge and discharge the insulating membranes. And that dotted line is suggesting the path that's taken as current capacitively couples through the cell membranes. Which path is taken depends very much on what AC frequency you use, and that will be the topic of another webinar dealing with the theory of ECIS. But for today, we want to look at the process of extravasation, the metastasis that comes about with cancer. And this cartoon drawing just points out some well-known events that have to take place when a cancer cell forms a secondary tumor. In figure 1, we see a group of cancerous cells. Some of them at step 2 have moved through the basement membrane, and ultimately at step 3 pass through the endothelial cells into the lumen of a capillary. They're carried downstream, which is step 4, and ultimately at step 5, they extravasate from the lumen through the endothelial layer and set up a new growth, a metastasis. This is what we want to try and study in tissue culture. Well, there had been work done before that showed that you could see in tissue culture a mimic of this in vitro process. And here we're looking at some figures from a paper by Garth Nicholson, where he showed that if you take and grow an endothelial cell layer to confluence and then challenge it with a cancerous cell, in this case a melanoma B16 cell, those cancer cells attach to the endothelium much as they would to the inside of the capillary and cause retraction of the endothelial cells and ultimately cell invasion through that monolayer in tissue culture. I have some more modern pictures of this process taken by Irene Newsham when she was at Baylor Medical School. And again, these are the flat cells are endothelial cells. The rounded cells are metastatic brain tumor cells. And you can see some of them have been false colored. And I'll just show you one of these blown up to show it making its way in the spaces between the cells. Well, this can be seen with electron microscopy, but we should be able to also see this electrically. Think of this. We have a layer of endothelial cells. Then we add metastatic cells. They attach, they cause retraction of the endothelial cells, ultimately moving to the substrate, which in this case would be the electrode. If we have current flowing, indicated by the red lines, this retraction opens up large spaces, and the current finds an easy path from the electrode through the cell layer. This should result in a resistance or an impedance drop, and also as more of the electrode is exposed, if one chooses to look at capacitance, there should be a striking increase in capacitance. To do this work, initially we worked with some researchers at Johns Hopkins who had the Dunning prostatic adenocarcinoma series. These are a bunch of sublines that were originally derived from the same tumor. They were mutagenized and passaged in tissue culture and then evaluated for their metastatic ability. We're going to look at 3 sublines. The G subline is weakly metastatic. The AT3 and MLL sublines are highly metastatic. Those are the cells we'll challenge an endothelial layer with. Our choice here will be to use HUVEC cells. But I just want to point out that HUVEC cells are a primary cell culture, and lots have different behavior. One thing we've found that's important when using these cells is to be very consistent, and having a gelatin layer or collagen layer on the electrode seems to be important. Here we're looking at 5 wells, 1 without cells, 2 that receive the HUVEC cells where we have not absorbed a specific protein. So the protein on those electrodes is presumably different proteins from the serum that's in the medium. The other 2 that come up high and stay high are gelatin-coated electrodes. This was an extreme example of how important coating the electrodes is, but it's something we always recommend. And so we're going to gelatin coat electrodes and then grow first a layer of HUVEC cells. We can use ECIS to follow the attachment and spreading of these cells, and by looking at the final resistive portion of the impedance, again, this is at 4,000 hertz, and also evaluating the fluctuations due to cell motion, we can verify that we have a confluent layer ready to be challenged with the metastatic cells. And here's the first result. Again, this is normalized resistance. We're dividing everything by its starting value. So all of these curves come out at 1. There are confluent layers present. But 2 of these wells are challenged either with the highly metastatic MLL cell line or the weakly metastatic G cell line. Notice, particularly with the highly metastatic one, there's a rapid drop in the resistance as we expected to see, which stays quite low throughout the experiment. The G subline requires more time before a less severe drop is seen. But we're adding as many metastatic cells as we have endothelial cells on the electrode, and so we also included, in addition to a control that received no cells, a control that received heat-killed MLL cells to show that the effect is biological. And actually, the control and heat-killed MLL cells on these small electrodes with all the micromotion are indistinguishable, and I really can't tell you which one is which, except that they behave the same way. So here we're seeing exactly what we expected to see based on the work that was done by Garth Nicholson and others, showing the breakdown in tissue culture of the endothelial layer. We wondered whether we could do this same effect by using medium that the MLL cells had conditioned, as this response might be due simply to proteases being released. But at least in our hands, when we did that experiment with conditioned medium, we saw no effect. And it's only upon the addition of the MLL cells themselves that we see the breakdown in the resistance due to the endothelial layer. Here's another example now using the AT3 highly metastatic sublines. And we do duplicate wells, duplicate sets of electrodes for that cell line, as well as for the G subline. And notice again, we see the G subline causing a weak drop in the resistance due to the HUVEC layer, whereas the AT3 cells requiring more time than the MLL, but ultimately they bring the resistance down to that of an open electrode, and notice all of the micromotion or movement seems to be gone. You may ask, why is that? After all, after these AT3 cells cause retraction of the endothelial cell, don't they go down on the electrode, and shouldn't we see a response from them? That would be a very good point, but in this case, the AT3 subline is essentially a rounded cell in tissue culture, and it's only cells that attach and spread that can effectively change the impedance of the electrodes. So what has happened here is the HUVEC cells have died, presumably undergoing apoptosis. The AT3 subline is alive but doesn't show up in the resistance measurement. Here's a look at several different human prostatic cancer cell lines that are all showing some effect on, again, a HUVEC layer. In the case of the Dunning series, we put the cells approximately in their right metastatic potential based on the severity of the drop in the impedance when the cells were added over the HUVEC layer. In this case, one might think the PTC1 was the most metastatic, but as I look at the literature, that is not the case, and actually it causes a bigger effect in vivo, but presumably not in vitro, but presumably not in vivo, just to point that out. Finally, we have a mathematical model that can be used to refine our understanding of what these cells are doing to the morphology of the endothelial cells. And we published this a number of years ago. The model shows that you can, instead of plotting time course changes in resistance or capacitance, you can plot time course changes in the problem of current getting through the paracellular space, which we call RB. It also can look at the effect of the cell layer on the problem of current getting out from beneath the cell in the space between the electrode and the basal membrane. And because current can capacitively couple through the cell membranes, the model also can give back the combined cell membrane capacitance. These three parameters as a function of time come out of the model, which simply requires that you monitor the impedance, the complex impedance, at a few frequencies, and then these parameters can be revealed. And here we see it, again, for a HUVEC cell being challenged with the AT3 highly metastatic subline. The top panel is the change in impedance, whereas the lower panel is looking at the three model parameters. And what is clear and not really unexpected is most of the change that we're seeing in the impedance is due to loss of barrier function. In other words, the spaces between the cell, the paracellular spaces, are less difficult to travel. What one would expect if the metastatic cells are actually causing separation of the HUVEC layer. Well, that's what I wanted to cover in this brief webinar. Our instruments that we used to make this measurement was the Z theta, which monitors complex impedance and allows modeling and so on. The instrument sits outside of the incubator with the laptop on it. In the incubator are stations which either hold 8-well or 96-well arrays. And I'd like to point out that we have a new instrument, too, that is less expensive, that will also probably be useful in making these same types of measurements. We call it the CP96. It monitors the degree of coverage of the electrode as a function of time. Here we're seeing cells responding to the addition of the MSO, but we believe this would work equally well looking at the metastatic behavior of different cancer cells. The nice thing is this is a relatively inexpensive machine and a turnkey package complete with everything you need, even a start of the arrays that are needed in a 96-well format is less than $17,500 in the United States. With both of these instruments, we always insist that you demo the instrument free of charge before you would decide to commit to purchase it. We want to make sure it works for your experiments before you make that commitment. Well, thank you for joining us. Our website is biophysics.com. There you can go, and if you click on publications, you'll find a list of different topics that ECIS can be used for. If you click on invasion, you'll see publications related to what we've been discussing today. I want to thank you for joining us. Remind you that the webinars are presented live on Tuesdays. Our next one coming up on June 2nd is dealing with automated cell migration measurement, where the instrument is used both to wound electrically and then follow the migration of cells as they heal the wound, equivalent to doing a scrape or scratch assay, but this is done all electrically with the door of the incubator closed, a neat application that you can consider using. Finally, here are some key contacts if you want to find out more about demos, pricing, and so on. Also, I am the last name there, Keese at biophysics.com. I'd be glad to hear from you, and if anyone would like copies of these slides, we're happy to furnish them so you can use them in your talks. We'd appreciate if you refer to the company when you use them. Well, that's what I wanted to cover today. Thanks for joining us, and we look forward to hearing from you. Take care. Bye-bye.